Forensic Lecture 7-1, The Role of a Forensic Toxicology. Toxicology is the process that detects and identifies the presence of drugs and poisons in body fluids, tissues, and organs. Their job does not just include crime labs, medical examiner offices. It also spreads into hospital labs and health facilities, identifying drug overdose, or monitoring drug intake within a hospital. In the US alone, drug manufacturers actually produce enough sedatives and antidepressants to provide every man, woman, child with 40 pills a year. So, this is part of the reason why drug abuse has become so prevalent here in the US. When we talk about a forensic toxicologist, they're actually limited to cases that pertain to violations of criminal laws with drugs. A major branch of forensic toxicology deals with measuring alcohol in the body. This is due to the fact that there's a high increase of traffic deaths that are associated with alcohol. 40% of all traffic deaths in the US are alcohol related. That's nearly 17,500 a year. And this is not including the 2 million injuries that require hospital treatment each year. Due to this increase, there is a need of forensic toxicologists to provide or devise a rapid and specific procedure for measuring intoxication that is safe and can apply to everyone. To understand how that works, we need to understand the metabolism of alcohol. All chemicals that actually enter into the body eventually are broken down. They're transformed into chemicals that are easier for the body to eliminate. This process is what we call metabolism. And there are three basic processes to metabolism: absorption, distribution, and elimination. So if we look at absorption and distribution of alcohol, it appears in the blood within minutes after consuming, and it slowly increases in concentration while it's being absorbed. Alcohol becomes evenly distributed throughout the watery portions of the body. So about 2/3 of the body's volume, it becomes evenly distributed with alcohol. When all of the alcohol has been absorbed, we have what's called the maximum alcohol level. And from that moment on, post absorption periods begin, which is the concentration slowly decreases until a zero level is reached. Factors that affect alcohol absorption rate are a number of things. Some of them are time taken to consume, so how fast they're drinking it, the alcohol content of the drink, the amount that was consumed, and then any food that was present in the stomach can all determine the rate at which alcohol is absorbed. Once maximum alcohol levels have been reached and it starts to disintegrate or break down, the elimination process happens. And this is accomplished through two processes: oxidation and excretion. Oxidation occurs when 95 to 98% of the alcohol will eventually be converted into carbon dioxide and water. And this mostly takes place in the liver by an enzyme that's called alcohol dehydrogenase. And it is first in the liver, first converted into acetaldehyde and then into acetic acid. The acetic acid is then oxidized into all parts of the body, into carbon dioxide and water. Excretion is any remaining unchanged alcohol, so that anywhere from 5%, 2 to 5%, is excreted in either the breath, the urine, or perspiration. The amount of alcohol in the breath they have found is directly proportional to the concentration in the blood. The elimination rate is going to vary by individual. So how fast the alcohol is processed and excreted is by individual, and it can actually vary up to 30% between individuals. So how the body actually eliminates it is we have to understand the anatomy of our circulation or circulatory system. So the circulatory system part of is the cardiovascular system, but it's the flow of blood. It includes the heart, which consists of 4 chambers, 2 atria, which receive blood from either the lung or the body, and 2 ventricle, which pump blood to the lungs or to the body. It also consists of arteries, which are blood vessels that carry blood away from the heart, veins, which are blood vessels that carry blood to the heart, and then a series of capillaries, which are tiny blood vessels that connect the arteries and the veins at the tissue level. And this is where the exchange of materials occur within the body, so between the body and the blood. There are 2 routes that the circulation takes. The systemic circulation, which carries blood from the heart to the tissues and back to the heart. And this actually contains a couple of subdivisions. The ones we'll talk about here is the hepatic portal circulation. And this is the travel of blood between the intestinal tract and the liver, which is what is oxidizing and taking care of the alcohol. And then the second circulatory route is pulmonary circulation, which carries blood to the lungs and back to the heart. So if we look at the circulation of alcohol, after consumption, alcohol is absorbed from the stomach and the small intestines into the bloodstream. It is then immediately carried to the liver, where the process of breaking down starts. From there, from the liver, it's then going to move to the heart and be pumped to the lungs, as well as to the other parts of the body. In the lungs, this process is also known as aeration, but what happens is the carbon dioxide and the alcohol that is in the blood leave the capillaries and enter into the alveoli. These are the air sacs of the lungs. Oxygen from the alveoli then enters into the capillaries. The carbon dioxide and alcohol is then exhaled through regular breathing. And as I had said before, there is significant evidence that verifies that the amount of alcohol in the breath that's exhaled is directly proportional to the blood alcohol concentration. So how much blood is saturating, or excuse me, how much alcohol is saturating the blood. From there, after the lungs, the blood with now the oxygen and still some alcohol enter back into the heart and is pumped out into the arteries and carried to all parts of the body. Any alcohol that still remains in the blood then moves into the tissues, and the blood returns back to the heart to begin the cycle again. And it'll continue like this until all of the blood has been processed and excreted. The amount of blood in the arterial blood, so that in the arteries, is higher than that of the venous blood, that in the veins during the absorption process. So it can be anywhere from 41% higher in the arteries 30 minutes after consumption. And so this process, being that they take the blood test from the venous blood, is actually in favor of the individual, being that if they're still in the absorption process, it's not going to read as high as it actually is. But eventually it will reach equilibrium. This is lecture 7-1. There's a quiz next class.